Okay, well, welcome back. This is lecture 12. Today we're going to be talking about stability and bifurcations. And I'm going to review the things that we covered last time at the end of class, because I think that was rushed a little bit. And I also think that it's worth hearing more than once. And then I'd like to do a little bit of an exercise based on what we talked about. I'll talk about one-dimensional bifurcations, and then I hope that we can begin to work on null clines and higher dimensional bifurcations towards the end of the class. And that will certainly occupy us for 2 weeks to get through all of that. And I have slides prepared, but I don't think we're going to get to the general theory of two-dimensional bifurcation, higher dimensional bifurcations today, although it's really not that different from the earlier material. So before we get too started on that, I was hoping to get a quick check-ins from people, just go around the room and hear where people are on their projects. We are in week 12 of the semester. That means, in principle, you have early versions of your project due in about 2 weeks. And so I wanted to make sure that we have everybody making progress and getting the support they need to help to make the projects work for you. And so maybe, Pedro, do you want to start and tell us where things are?
Yep. Yes. So while I'm opening everything here, I'll explain the situation. So it's a fairly complicated network, and if I base my project on their equations, it's gonna go wrong. The ODEs are consistent. The ODEs are very consistent, but the diagrams are not. And
you want to remind people what you're working on?
The reactions are not too. Yeah, so I'm working on replicating a model of innate immune response to viral infection. Let me go down to my project. So this is the network. There are a few proteins that respond to viral infection. So for example, TNF alpha, interferon alpha, interferon beta. And there is the STAT pathway that controls signaling from interferon, and they all get regulated by RNA reactions. So I started from the ODEs because the reactions and the diagrams are inconsistent. They don't make sense. So I'm using the ODEs. So basically I'm transcribing the ODEs as rate equations that I can copy and paste into the Tellurium model. And till now I've done that to all equations they showed. So I didn't go as far as P, V, and W proteins from the Nipah virus interacting with this network, because I will, in the future, focus on influenza, which does not have P, V, and W. But I will surely take a look on how they do it later. So they have the production of interferon beta mRNA, which is a basal production, a linear production. Here they are not using Michaelis-Menten, and a decay. They use the Michaelis-Menten to produce the interferon, and this interferon is immediately released in the environment. So they are modeling the production and the secretion at the same time in this equation. So they are modeling the infection in dendritic cells in an environment. I don't know exactly what environment it is, but there is a volume. This environment has a volume, and they know the number of cells. So this interferon in the environment has to be normalized by the number of cells in the environment and the volume of the environment. This method here for the TJ SOX sensing part of the interference, they use a technique that I think it's worth showing you guys, which is basically writing the rate equations and saying it's 0. So basically they're assuming these equations here are much faster than the rest of the network. So if they are much faster, they reach the steady state much faster. So d by dt of each of these molecules is 0. If you do this, you can actually derive the algebraic relation between the components and just use them as the value of the component you are interested in. So in this case, they are interested in the levels of TJ, which is TJ is the binding site to interferon. And when they are phosphorylated, they trigger stat the stat simulink pathway. So they are only interested in the TJ phosphorylated. So they can derive the TJ star here as a function of everything else. So there is no ODEs here, which is nice, doing this interesting assumption. And I've done this, yeah, so they do this also for the stat pathway. And I basically went through all equations and wrote them one by one as rate equations that I can later on just copy and paste and to the Tellurium code. Yeah. So that's all I have. So next step for me is writing the Tellurium model.
That's fine. I mean, one small thing is when you've got these things written as assignments, not as rate equations, right? So I guess you could always put that into a rate equation, say DC, right? The DCCL5M equals rate underscore CCL5M. Is that what you're planning on doing?
Can I do that?
Well, because you have things written as rate of something colon equals, which is an assignment, not a ODE.
Yeah, it's not an ODE. So what I plan to do is this. Let me write it down. What I plan to do is CCL5M rate CCL5.
Right. That's what I was expecting. I think that will work. I haven't tried it, but that should work. Yes. That should be fine.
I mean there's no way around. I mean, I would have to reconstruct all the network from the ODEs before implementing on Tellurium.
Well,
or I do this, or I write the rates
or you just write CCL5M arrow CCL5M semicolon and then the rate, right? It's equivalent to it.
But in this case, shouldn't the mass be conserved because I lose one CCL5 and gain one CCL5?
It doesn't change. The things you've written are equivalent. Well, we could talk about that separately, but I don't want to take too much time today. I just wanted to make sure that looks great. that you got that. That's already a lot of progress. So it'll be exciting to see you actually start running those simulations and seeing what they do. Okay, great. Any other comments from the class on that? Okay. Elmar, do you want to tell us what you've got going?
Yeah. Okay. Well, I have not yet transferred my project to Colab, and I have nothing directly to show. So I think this time I just have to pass.
Okay, that's all right. If there's anything we can do to help you out, let us know, and we'll do our best to be supportive.
Yes.
Okay. Ibrahim.
Yeah, I can go ahead real quick, I guess. Yeah, so just a reminder, my project, just make sure everything's running. My project has been to model predator-prey interactions. And one of the, I guess, so earlier I had, so basically what I've been doing is kind of been trying to figure out which parts of the predator-prey model I want to add to my rate equations. So kind of I've just been compiling different sections from different papers, trying to find which things I can just add by doing like a plus or minus, because those are the easiest ones to add to the rate equations. So that's what I've been doing. And then I've also been trying to use the code that we worked on last class to get these phase plots going.
Well, today should be good. If we get through the material I planned, we're going to do the simple Lotka-Volterra equations, which are the most simple predator-prey model. And we'll walk you through all the steps of doing that. So
Even better.
maybe that'll be helpful. Maybe that'll be helpful for one of the reasons I wanted to do these phase plot lectures this year was because it seemed to me that a lot of people were doing things that would benefit from that. So I wanted to make sure that we get through that material for you. So I can't promise that everything will be transparent after today, but at least we'll have a little more practice on that.
Yeah, well, I mean, basically what's happening for me is because I compiled these kind of like additions to the rate equations across multiple papers, I kind of am playing around with all these parameters, trying to figure out which one works. I'll probably do a parameter scan now that I think about it, to visualize it in over time first.
Mm-hmm.
And then I'll probably do this after. That's probably a better way to start.
Yeah, because those phase plots take a little while to run, it's useful to get a sense of what the ranges are of the variables. In other words, are they between 0 and 1, 0 and 1,000, and so on. Helps you get oriented a little bit. And
Yeah.
again, I'll show you a little bit today how to maybe some ways of thinking about those kind of problems. So that's great. Anything else you wanted to talk about today on that?
No, this is kind of where I'm at right now. I mean, my project in the end is going to be basically allowing the user to manipulate some of these parameters, and then plotting both the predator versus time, prey population versus time, and then also these phase plots. And then kind of walking it through educationally. So this is kind of the step I'm on right now, I guess. This is basically all I had to show as well.
Okay. I would encourage people, some years I've given a lecture on it. I don't know whether it's really worth taking class time for it, which is if you're going to build these notebooks that are going to be maximally useful for a user, especially a naive user, you probably want them to be able to interact with some graphical tools. And so Colab supports IPy widgets for things like sliders, dialog boxes, and so on. And so I encourage people to look up a basic IPy widgets tutorial. That's how that's the basic way you set up the user interface, so that the user doesn't have to type and rerun the cells, but actually the cells run in the because Jupyter Notebooks is designed so that the cells run server side in the background, and the user interacts with them via a widget interface. And so that'll make if you can get that set up properly, it makes it a lot easier for a user to interact with the code. I can maybe later in the semester talk a little bit about that, but given the timing, I don't want to take too much time for that. Once you've done one IPy widget, they're all pretty much the same. But it is event-based programming in the sense that your simulation runs in the background, and then when somebody manipulates the widget, it reruns the simulation in response to the user input. And so you have to set up a callback structure. Once you've done it once, it's all pretty much the same. And I think I may have some examples. The good thing is that in NanoHub, they were fairly limited. They didn't let you have all of the IPy widgets, whereas in Colab, you can use any of them. So you're not limited to that. And that's the kind of thing where I encourage you all to put your heads together and help each other out to get started with that. It's not hard. In an hour of reading, you can figure it out. But if you haven't done it before, it takes a little bit of playing around. Okay, so we've heard from Elmer, Ibrahim, Pedro, William.
Yeah, so I've been struggling with the IPy widgets thing for a long time probably because, and I didn't think about this, I'm doing it in VS Code right now, and it probably doesn't work in VS Code, right?
I don't know whether they'll work properly in VS Code.
I run it. It's correct, but it just infinitely runs, or there's like a weird error. I don't know. So, I mean, what I've been working on is I've been reading a lot of papers and writing notes to make sure that I understand, because when I was going back and reading other people's projects, they seem to know a lot more about what they are working on, what they worked on relative compared to what I know or what I knew about what I was working on. So I think I needed to and their descriptions were very long and detailed and helpful. So I'm trying to make that part helpful. And I've also modeled one or two different models for P53 DNA damage. But I think my plan this week is to try to put more code together and get the Colab notebook started with. I'm also struggling with, I don't know why, but yeah, I think I need to transfer everything I have on VS Code to Colab and then implement the interact features. And I saw on some notebooks that and I think in the assignment, do you want us to gradually increase the complexity of the parameters that they can change so it's easier to understand? You understand my question?
Yes. I mean, you should think about this as something that's going to be put out there that people will look at. And so your name will be on it, and it will be public.
Yeah, well,
so you want it to be something, one, that you're happy with, but two, if you think about it, again, I don't know. That's why I shared with you some of the older projects, some of which I think worked very well, some of which were less successful. Typically, what you want it's a little bit like a term paper, but the figures are alive, right? You want an introduction to the biology, an introduction to the mathematics. Doesn't have to be very long, a page or two maybe. And then you want to have a simulation where this user can control some key parameters and see how the simulation responds. And if you can do that all with a single simulation, that's fine. But typically, just the way we try in the class, we'll start out with a simpler example, and then we'll try to apply what we learned from the simpler example to a more complicated one. Typically, these notebooks will have maybe 3 or 4 examples, and typically the first ones are simpler to introduce the ideas, and then gradually building it up. If you think about it, you don't have to think about it this way, but if you think about it, for example, suppose that you were going to take your notebook and some professor were going to use it as a section of a class to introduce an idea and teach some concepts. How would you organize it so that it worked like that, so that you're helping teach people who don't know this material before? And I think Hayden is very good at trying to break things down that way. So if you have questions about how to organize it, I think I definitely reach out to Hayden for some advice, and I'm happy to help you too.
Okay.
One of the reasons that I really wanted people to have a draft version of their simulations ready 2 weeks before the last class is so that everybody could present their work and then everyone could critique it. Historically, what we've done is we've had each person or each group has picked another group, and you do a full presentation, an hour or 2 hours, to that other group, and then they write a detailed critique. This is clear, this is not clear, this worked, this didn't work. And then you have a week or 2 to go back and fix things so that it's not a first draft that gets published. Since it's something that's going to have your name on it and be out in the public, you probably want to have something that you want take the time to revise it so that it's something that people can appreciate.
Got it. Okay.
I mean, I don't want to make it sound too intimidating. It doesn't have to be very complicated, but you do want it to
I think I was
clear.
Yeah. No, sorry. I was intimidated by the length of some of the projects that I saw. It was they had a bunch of simulations and then they tried it with real life data, which makes sense. And then they were like, oh, let's do more. Let's do double. It was like a COVID vaccine. Let's do like, what if there was double vaccines or something? What if there's blah, blah, blah? And then they did a whole thing about that. And I mean, I got bored like halfway through. So
well, okay. So then your critique of those was that they were too long and not exciting enough.
Yeah, I guess. Yeah.
So that's okay. I mean, there isn't, I mean, if you turn in 2 lines and one cell that says I had a project and then the simulation consists of drawing a horizontal line, that's probably not enough. It doesn't have to be a PhD thesis either. There has to be some.
I also had a more specific question.
Sure.
About, I know you talked about compartments, but what's the point of compartments like cytoplasm, nucleus, but what's the point of specifying those in like a model per se? Is it just for organization, or can you specify like is it for the rate of transfer of things in between compartments?
Yes. So certain, I mean, in the cell, RNA synthesis happens in the nucleus, but protein synthesis happens in the cytoplasm. And so if you were really going to be modeling RNA synthesis and protein synthesis, you'd have a step where you had the production of RNA in the nucleus, and then the transport of the RNA from the nucleus to the cytoplasm, and then the translation of the RNA into protein by the ribosomes. And so you might have quite different chemical reactions in the nuclear compartment from those that are occurring in the cytoplasmic compartment. If you had a model of mitochondria. Mitochondria are quite isolated in many ways from the rest of the cell. And so they have very different chemical reactions going on from the rest of the cell. They even have a slightly different genetic code from the rest of the cell. And the same sorts of things happen if you're doing models of organs. The chemical reactions happening in your liver are very different from the ones happening in your kidneys. And so if your compartments represented organ systems, you might have very different sets of equations going on inside of each one of those because they represent different reactions. The other one, which I actually saw in Pedro's code, I think, was that in that case, they were actually counting the number of virions. And so typically our reactions are based on concentrations, not amounts of chemical. I've said at the beginning, I'd say amounts or concentration, amounts or concentration. But you have to convert concentrations to amounts by knowing the volume. And if you're going to do transport models, or you actually want to have amounts, then you have to know the volume of each box in which your reactions are occurring. Because reaction rates don't depend on amounts, they depend on concentrations. So you have to be able to convert amounts to concentration with a volume. And so situations like that, you also need compartments because the volume of the compartment then becomes an important parameter in the simulation. So you don't all, I mean, there are plenty of models which don't need compartments, but it is a convenient concept if you have either regions that have fundamentally very different biochemistry or regions where you need to actually count how many molecules you have, in which case you need to be able to convert concentrations back and forth into amounts. Does that help at all for that?
Yeah, that was very helpful.
Okay, I don't know, Alex. You're not officially, but you're working on projects. Do you want to say anything?
Probably not. I don't really have much to talk about right now. Maybe.
Okay. I don't want to put you on the spot, but since everyone else said something, I thought I'd give you the opportunity if you felt like. Okay. Okay. All right. Great. Well, thank you. And again, I'm not. My goal here is not to make things hard for people, but I do want to make sure that people don't get too far behind, and especially if you're stuck, that you feel you can reach out and get help. Hayden and I are both available. Giuliano was just down here. I don't know how available Giuliano is, but he's a big expert in this kind of modeling. So he was the TA for this course for something like 4 years in a row. So he is around on campus.
Everybody's seldom here.
Okay. All right. So everybody see this okay? Just make sure I've screen shared all right. And you can hear me. Good. All right, so I want to come back to stability and fixed points. And I haven't labeled these as review, but we're going to start basically from where we were last time. I tried to clarify some of these slides a little bit. So we've heard the concept, and we've seen many times now, the concept of a fixed point or steady state of some set of dynamical equations, which is the values you achieve when you run a simulation for a long time. And in principle, we need to know the concentrations of each species in our system to be able to determine whether we're at a fixed point or steady state. But if we write all of our concentrations as a big vector, x1, x2, x3, x4, x5, however many variables we have, and we aggregate our rate equations into a big vector where f vector represents all of the rates put together, then we are a fixed point or a steady state when that rate f of the fixed point value x star is 0. And it has to be 0 in every rate equation. All of our rate equations have to be 0. And I realized that the notation when we start writing things for vector forms looks a little bit different from what we're used to writing, because normally each chemical species would have a name like ATP, FGF, something else. When we're going to write it as a vector, they all wind up being X1, X2, X3, X4, which is less informative. And the question is, when is a given fixed point stable? In which case, by which we mean if we move our concentrations just a little bit away from our fixed concentration, do we come back to where we started, or do we move further away from it? If we move further away, we're unstable. If we come back, we're stable. Now, the case of oscillations is a little bit different, and we may get to oscillatory solutions today. Otherwise, it'll be the next time we get together. And we have this x is a function of lambda, a function of the parameters of the problem, because as we change the parameters, the fixed points themselves will move around, and that's really what bifurcation analysis is about. And just because last week Elmer said that this notation was a little bit unfamiliar, I'll remind you that the vertical bar, that vertical line, indicates where the derivative is being evaluated. If I write dx by dt, then that doesn't tell me what value of x I need to use to evaluate f of x star. So that's what that's for. Oops, sorry. So let's start out by looking at the simplest possible case, which is we only have one chemical species. So we're now talking about a one-dimensional rate equation. So now we don't have any vectors anymore. So we have d by dt of x is some function of x. And x star is a fixed point if f on the right-hand side, the rate as a function of x star, is 0. And so I've sketched that here. I've written f of x versus x. And anytime f of x crosses the 0 line, we have a fixed point. And so in the picture I've shown here, we have 3 fixed points, one at 0, one at a half, and one at 0.9 something or other. So from a graphical point of view, this is pretty straightforward. I plot f of x, and any place f of x is 0, I have a fixed point. And again, x star is a stable fixed point if I move a little bit away from my initial value of x star, and I come back, and unstable if I move further away. And so the example, and this is actually from the class that we've done, here I've got time series, and I see that if I start out with values of x between 0 and about 0.45, they decrease and come to a steady state value, I don't know, about 0.1. So that 0.1 is clearly a stable fixed point. For values between 0.5 and any larger number, 2, they come down and reach a value of about 1. So that's also a stable fixed point. What's a little harder to see is that in between the values that go up and the values that go down, there's a fixed point of around 0.5, and that's clearly unstable. If I start just a little bit above it, I go to the fixed point above it. If I start a little bit below it, I go to the fixed point below it. And so that's an unstable fixed point. And that actually corresponds pretty closely to what I have. In fact, it corresponds exactly to what I have on the top, because the time series trajectories actually are derived from that particular function. So that is exactly that. Now, we have this words near. We have to start near x star. And there's some mathematical language that says that there's some range, delta, such that for all points within that range of x star, you wind up at x star, you wind up coming back to x star. That's not so interesting in one dimension, but if you go to higher dimensions, that existence of that range is not so obvious. And we'll start with x star plus some initial perturbation. And if you're a physicist, this is called perturbation theory, perturbation analysis. Mathematicians will call this linear stability analysis. Same thing. And so we're going to start out with x star plus epsilon of 0. And we want to know when we're stable or unstable. And so what
Second, mathematics is linear stability. What was the perturbation?
Elmer asks me to repeat what I just said. I said that physicists tend to call this perturbation theory because we start with x star and we add a small perturbation epsilon to it and see what happens. And mathematicians will call this linear stability analysis. And the reason it's linear stability analysis is that we are restricted to epsilon being small. And what small means we'll come to in a few minutes. So we're now going to start with an initial value of x, which is x star, a fixed point, plus epsilon 0. And we want to find out what x of t is. And that's going to be written as x star, which doesn't change in time, plus epsilon, our perturbation, which does change in time. And if epsilon goes to 0 as time increases, that is, we come back where we started, then we're stable. So the way we're going to do this is we're just going to take our dynamical equation. We only have one of them. dx by dt is f of x. And we'll plug in x star plus epsilon. So we haven't done much there. And on the left-hand side, we have now d by dt of x star plus epsilon, which we can separate into d by dt of x star plus d by dt of epsilon. But x star is a constant, so its derivative is 0. So now we have d by dt of epsilon is equal to f of x star plus epsilon. So we're turning a rate equation for x into a rate equation for the perturbation for epsilon. And now I think everybody has probably run into at some point in their lives a Taylor expansion. This is something one typically does, I think, in high school, but it may be so long ago that you've sort of forgotten how they work. And there aren't that many things that I would say one has to remember in mathematics for engineering or science, but there are a few, one of which is that d by dt of x equals x is an exponential. That is something one has to have in one's blood. The first order Taylor expansion is also something that one really does need to have a little bit in your blood. But, and one can actually derive the Taylor expansion. I'm not going to do it here. If you want to remind yourself of the derivation, Wikipedia has it. There are plenty of places you can find it. But it has a nice form, which is that if f is a continuous function, a continuous differentiable function, I should say, and it's a function of x plus some small perturbation, I can write f of x plus a small perturbation in the form of f of x plus something that depends on the perturbation. And so you can ignore the d by dt of epsilon because that is not a Taylor expansion. We're just expanding the right-hand side. And we're saying that f of x plus epsilon is equal to f of x plus, and now the first order term is df by dx, the slope of f, evaluated at x times epsilon. So it's a linear change in f as a function of x. And then the next order term in the Taylor expansion is 1/2 times the second derivative of f, also valued at x, times epsilon squared. And then the next one would be 1/6 the third derivative of f evaluated x star times epsilon cubed, and going up in higher orders. The reason that we're going to call this linear stability analysis is that we're only going to look at the second term, which is the one that's linear in epsilon. Now, the first thing that we can notice is that f of x star, by definition, is 0, because we've said that x star is a fixed point of f. And so just the way the derivative of x star disappeared on the left, the f of x star now disappears on the right. And so we're left with d by dt of epsilon is equal to df by dx evaluated at x star. That's just a number. That's the slope of f at x star times epsilon. And then potentially we need to look at terms in the order of epsilon squared, epsilon cubed, and so on. When we say that epsilon is small, what we mean is that that first term, which is linear in epsilon, is much bigger than the second term that goes like epsilon squared. So if epsilon's a number between 0 and 1, epsilon is always going to be bigger than epsilon squared. If epsilon were 0.1, epsilon squared would be the order of 100th. And so we have to decide what small enough means, but it certainly means that epsilon is less than 1 in this case. And so we're going to do what's called linearization, which is to neglect terms that go like epsilon squared, epsilon cubed, and so on. Now, if df by dx evaluated at x star was 0, then that first term is 0, and we absolutely cannot neglect the higher order terms. And so we need to assume that the derivative of f is not 0 at x star. If it is, then we have to do other things. So we can't linearize if that's the case. And if we can linearize, then we have something very simple. The derivative of epsilon with respect to time is epsilon times the slope of f evaluated at x star. Again, d by dx of f of x evaluated at x star looks very complicated, but remember that's just the slope of f when it crosses the 0 line, and it's just a constant, it's just a number. And so what we have is d by dt of epsilon is equal to some number times epsilon. And so remember, that was one of the things I said, it's one of the 2 or 3 mathematical things you have to have in your bones. When you see d by dt of epsilon is equal to a constant times epsilon, we know that the solution is exponential. And so now we can write epsilon of t is equal to our initial value epsilon 0, times e to the time times the slope d by dx of f of x evaluated at x star. Okay, are there any questions about that? Going over it the second time. This linearization and stability analysis is something that one does again and again and again in physics and in engineering and in mathematics. And but depending on your background, it may be something that you've done more frequently, less frequently. So this is as good a place as any to get familiar with. So if the slope, d by dx of f of x, is positive, then e to the t times that is an increasing function, which means we're diverging. So the stability of the fixed point is given in a very simple way. If the slope of f at the fixed point is negative, we have a stable fixed point. If the slope is positive at the fixed point, we have an unstable fixed point. And if the slope happens to be 0, we have what's called a marginally stable fixed point, and then we can't linearize. And so we don't really know what's going on. We have to look at other things. And let's look at this graphically now. Suppose that we have f, we plot f of x versus x. Whenever it crosses the 0 line, we have a fixed point. If it crosses the 0 line going down, the fixed point is stable. If it crosses the 0 line going up, the fixed point is unstable. And if it has slope of 0 when it touches the x-axis, then we have a marginally stable fixed point. And so if we plot f of x versus x, we can immediately read off the values of the fixed points wherever it crosses the 0 line, and the stability. If it crosses the 0 line going down, it's stable. If it crosses the 0 line going up, it's unstable. So we don't really have to do any math. We just do the plot and look at it. So in the example that I showed here, I plotted f of x versus x. It crosses 0 3 times at 0, a half, and 0.9. The first time it crosses it going down, so it's stable. In the middle, it's going up, so it's unstable. Third place, it's going down again, so it's stable. So I have two stable fixed points and an unstable fixed point. If f of x is continuous and differentiable, you'll notice that the fixed points are going to have to come in pairs. Because my line, I couldn't have two consecutive stable fixed points, because I can't go down and cross 0 twice without going up again. If f of x is not required to be continuous, then I could have two stable fixed points next to each other. And so most of the theory that I'm going to be talking about is going to assume that our rate equations are continuous and differentiable. If you allow discontinuities in the rate equations, jumps in the rate equations, then some of the things that we're going to talk about here aren't necessarily going to be true. In general, in most situations, rates change continuously when I change parameters. But there certainly are situations where they don't. And if you deal with discontinuous rate functions, then the dynamics can be a little bit more complicated. Okay, any questions about that? Again, I'm trying to complement the mathematics with some pictures to make it a little bit less abstract.
I've got this more, fixed point because you would have this. Can they hear me?
Yeah, go ahead. I'll hear it. Let me mute. And I'll do it. Let you talk.
Okay, now I do. My question is, when you have a tangent, maybe I did the camera on. So when you have the tangent that comes down to the 0 line, my finger's the 0 line. So now first you don't touch, but then there would be parameters that you can shift the tangent, and at one point it just touches, and this is the marginal one, I guess. And if you would go deeper down, then you have a stable and the unstable fixed point. Is this the idea?
So that is exactly the idea, and it's in fact going to be the next slide.
Oh, great.
So Elmer beat me to my next slide. Okay.
So my follow-up question is that how do you shift the function? How will you shift the tangent function? Is this, how do you know which parameter to tune that you can do that?
Okay, so let me come back to this. So Elmer asks, how do we know which parameters to tune? Well, the answer is that when we write f as a function of potentially many parameters, we have to plot f of those parameters. And as we change each parameter, we have to look at what happens. And I'm going to walk you through two examples of that now. I can't tell you every possibility. I can tell you every possibility of bifurcation because there are only four for one-dimensional maps. There are only four possible bifurcations for continuous differentiable maps. But Elmer is asking exactly the questions that I was hoping somebody would ask going forward. So we're now going to consider what happens when we change that lambda. This is a parameter that has some effect on the function. And the first definition that we're going to have will seem perhaps a little bit abstract, but I hope that when we go through some examples, it'll be clearer what's going on. So I'll give you the definition first, which is the equations d by dt of x of equal to f, some rate of lambda of x. And I've got the vectors again, because this is true for any number of dimensions. It's not only true for one dimension. Have a bifurcation at a critical value of parameter, lambda c, if and only if the number or stability of the fixed points for lambda a little bit bigger than lambda LC differ from the number on the other side. So if I increase or decrease lambda through lambda critical, either the number of fixed points change or a fixed point that was stable becomes unstable. There's some change in the identity of the fixed points. Now, it happens also that if f of lambda is differentiable in both lambda and x, then, as Elmer said, the slope of f at the fixed point has to be 0 at the bifurcation, at the critical value of lambda. So let's actually now look at an example of that. So suppose that I have a curve that's a decreasing function. You could imagine, say, a quadratic. And I'm going to gradually increase the value of lambda. And as I increase lambda, the position of that curve moves down. F of lambda decreases. So maybe I should write f of x minus lambda would do that. So for my lambda 1, I'm up at the top here. And I have no fixed point at all. My f curve doesn't cross the x-axis at all. No fixed point. I now increase lambda a little bit. It moves down. It's still not crossing the x-axis. I get to my critical value, and it just touches the x-axis. It's tangent. And that's a marginally stable fixed point. And as I've said before, to really understand what's going on at the marginally stable case, I have to do a little bit more math than we're going to do here. But in general, I'm not going to worry too much about what happens in that one specific value. I'm now going to keep increasing lambda. And now my curve, my f curve, dives below the x-axis. And so I now have two crossings, one going down on the left, which means I've got a stable fixed point, and one coming up on the right, which is unstable. And if I keep increasing lambda and pushing my line further down, those two fixed points move away from each other. So if I saw this as a little bit of a video, suppose I had a video where I gradually increased lambda, I'd see along my x-axis, I'd see no fixed point, no fixed point, no fixed point, no fixed point. And then I create a marginally stable fixed point, And then I see that marginally unstable fixed point split into a stable and an unstable fixed point that gradually move away from each other as I increase lambda. And that's the bifurcation, yes. Now, the other way that this can happen is, in that case, I have a loop that's coming down and hitting the x-axis from above. I could have had it come up and hit it from below, too. The other way I can think about it is, suppose that I have a curve that's crossing the x-axis already. So here's f of lambda, and it's crossing and it's decreasing. So I have a stable fixed point. And now, as I increase lambda, I begin to get a kink. Imagine that I have, say, a cubic function that is pulling my line to make it look a little bit like a chair. As I increase lambda more, the values on the left are coming down. The values on the right are coming up. There'll be some critical point again where just at the fixed point, I'm tangent to my x-axis. And so my fixed point goes from being stable to being marginally stable. And now I keep twisting it so that my f of x dives below the x-axis on the left and above on the right. And so now I have a cubic shaped function, a S-shaped function, that's decreasing on the left. So it's created a stable fixed point on the left. My fixed point that was stable to begin with is now unstable in the middle. And I have a second stable fixed point on the right. And so this is the other way that I can get the creation of fixed points. And again, I've created a pair. I've gone from, in the first case, I went from no fixed point to one stable and one unstable fixed point. Here I went from one stable fixed point to two stable and one unstable fixed points. And it's a general theorem that the sum, if you add up the number of stable minus the number of unstable fixed points, that number doesn't change when you change your parameters. The bifurcations leave that number unchanged. Again, that's only true if you have differentiable functions, continuous differentiable functions. All right.
Wait, sorry, I had a question.
Sure.
So wait, just to make sure, on the last slide, the text was incorrect. It said one stable, two unstable, but that's two stable and one unstable.
Ah, thank you for catching
Right?
that. Yes, that's right.
Okay,
Thank you.
Okay.
Thank you for catching that. I've noticed a couple of typos in these slides. That one I missed. Thank you. Okay. So there are a couple of kinds of bifurcation that you can have. We've shown cases here, two cases where we had fixed points that started out stable and became, but no fixed point generated a pair of stable and unstable. We showed one where an initially stable fixed point created two stable and one unstable. If I look more locally, if my F line is changing slope, it can go from being stable to being unstable, or vice versa. And so these are the possibilities. So I could either have the identity of a fixed point change, I can create or destroy fixed points, or I can have a stable fixed point become unstable or an unstable one become stable. And so now let's come back to the example that we did a couple of weeks ago, which was auto-activation. And this is really, I think, the nicest simple example for bifurcations. And we actually did do the bifurcation plots in class, although we did them before we really talked about what they meant. So now I want to come back and do them again, maybe with a little bit more insight into what those bifurcations mean. So here we have a species that promotes its own production. As before, we're going to interpret this as a gene regulatory network. So A promotes the production of A. We have to remember, as always, that when we write a gene regulatory network, we have to assume decay of the species A. And we're also going to assume a basal rate of transcription for this molecular species. We could include a messenger RNA step. We're not going to do that today, although it's interesting to do that. It doesn't change things very much, but it wouldn't be a bad homework problem to try to redo this with that extra messenger RNA step. And so now we're going to write a Hill function for the rate of production of A as a function of the concentration of A. And I'm going to scale my Hill function by a half so that the value of A is typically between 0 and 1. And so I'll write the rate of production of A is a basal transcription rate, K basal, plus V max, A to the H over 0.5 to the H plus A to the H. And the choice of 0.5 as my scaling factor doesn't really make any difference. I could have picked 1, I could have picked 10, that wouldn't change anything. We can also simplify this by essentially measuring our time scale in convenient units. And often we'll measure things in terms of the decay time. So we'll set the decay rate equal to 1. But in this case, I'm going to set Vmax equal to 1. So that shows us that there actually are only 2 free parameters in this problem. If I put Vmax in as well, I would find that I actually was over specifying them. And so my final form for this, which we've worked with already a couple of weeks ago, is the rate of production of A is equal to basal transcription rate K basal plus A to the H over 0.5 to the H plus A to the H, increasing Hill function, minus decay rate times A. And so I have 2 parameters in the problem, K basal and D1. And let's look at the right-hand side of this, the rate, call it f of A. Well, suppose K basal were 0. If K basal is 0, I have A to the H over something minus D1A. So clearly 0 is a root of this. If I plug in 0 and K basal 0, I get 0. So that's a fixed point at 0. If I plot it, I get a curve that looks like this. This is still keeping K basal equal to 0. I have a fixed point around a half and a fixed point at here I've set D. Let's let the decay rate be 1. Makes life a little bit easier. So D1 be 1. Well, if A is equal to a half, then a half over a half to the H plus a half to the H is a half, minus half is 0. So a half is clearly a fixed point. The large one, which is close to 1, is not quite so obvious. That one is something you have to play with to find. But I have a third fixed point around a little bit below 1. In this case, it's around 0.9. And so I actually can analytically find the 2 solutions, 0 and a half fixed points by hand. And then that third one I have to evaluate numerically. Now, stability, I know from just the shape of the curve that 0 and 0.9 are stable. A half is unstable. And so that's what my final result is. And we've done this, again, we did this in class a couple of weeks ago. So then what we did was we plotted for different initial values of A. We plotted the time series. And we saw that if the value of A was a half, it stayed a half. If we started even a little bit above a half, it went up to 0.9. If we started even a little bit below a half, it went down to 0. So that confirmed our stability and identity of our fixed points. So now let's see what happens when we change k-Basel. When we change k-Basel, we're adding a constant to our entire function. And so that's taking our curve and sliding it up. It's moving it upwards. So if I move my curve up, what happens to my crossing points? My initial stable fixed point was at 0. I slide the curve up, the crossing point moves to the right, so it gets bigger. So as I move k-Basel to be bigger than 0, that lowest fixed point stays stable, but its value goes from 0 to being above 0. What about the one that was just below 1? It also moves to the right and stays stable. What about the unstable fixed point? The unstable fixed point, as I slide my curve up, my unstable fixed point slides to the left. And so the distance between the first stable fixed point, the small one, and the unstable fixed point gets smaller as I increase k-Basel. And the distance between the unstable fixed point and the larger stable fixed point goes up as I increase k-Basel. So I've got this sigmoidal curve. I'm just sliding it up and down and asking, where are the crossings with the 0 line? If I make k-Basel to be too big, specifically at a value of 0.2, that unstable and that stable fixed point collide. That lower branch of my curve, that dip of my curve, doesn't cross the x-axis anymore. And so the two lower stable and unstable fixed points collide, and we call them, say they annihilate each other. On the other hand, that stable fixed point around 1 never disappears, no matter how much I move up or down. Well, move up, it's there. If I made k-Basel negative, I could get it to disappear. Then only the lower value would stay. But it's not biologically meaningful. I can't have a negative transcription rate. Okay, so does that, I want people to look at this picture and imagine that blue line being slid up and down and think about where those fixed points are moving, left or right, as I move it up and down. I guess it would be nicer to have a little animation of this, but I hope that the diagram is helpful. So you really can think, in a sense, very geometrically about what's going on. We're solving a little bit of algebra here, but fundamentally we're thinking about how curves move and how intersection points change as things move. All right. So if I increase k-Basel, 0.1, I still have two stable fixed points. Notice that the value of the fixed points have moved up a little bit. Instead of 0.9, it's now 1 point something. Instead of 0, it's 0.1 something. And now I do k-Basel equal to 0.2. So that that fixed point is almost there. I'm almost touching the zero line, but not quite. And so now for any initial value, I wind up on that upper stable fixed point. But you'll notice it takes quite a while. If I start for a very small value of A, the value of A goes up pretty fast, and then it sits near 0.2 for a while, and then it jumps up. And that's called a ghost, because I almost have a fixed point there, but not quite. And that ghost of the fixed point means that the derivative of A is small, not quite, it never goes quite to zero, so I never stop, but it's slow. I move through that region quite slowly. And so I can tell in that lower one that there was a fixed point there, even though it's disappeared. Because I see myself sitting in that region where there used to be a fixed point for quite a while.
So, how does now this lower one compare to the curve that we see? Where would we be? Is now a march?
So Elmer says, where is this 0.2 place we're getting stuck? That 0.2 is getting stuck is exactly the minimum of the blue line, where it's almost touching the x-axis.
Then it disappears in
Right.
the only one.
Right, that's right.
And then another question. These A2 function of A diagrams, are they called nonlinear maps?
So Elmer asks, is f of A versus A a map? So there are two classic ways of doing dynamical systems, one of which is rate equations, like the ones we're doing, dA by dt equals some function of A, which are continuous in time. The other one are what are called iterated maps, where I ask the question, for example, if the population of flies is such and such this year, what is it like next year?
Okay.
And in that case, I'd say A sub n plus 1 is f of A of n. Those two kinds of step-by-step map versus continuous dynamics are related to each other, but they're not quite the same. And the stability analysis and the bifurcations and everything else are slightly different. And if I were teaching a mathematical biology course, we would do both the iterated map theory and the dynamical systems theory. But in chemical reactions, we always have continuous change in time. And so we're going to focus on that today.
Can I just ask one follow-up? If I have a function and I do it iteratively and non-continuous, and I have a bifurcation point, will I have this bifurcation point as well in the non-discrete function?
So Elmer asks, if you have a function f of A, and you had it as an iterated map where you had A sub n plus 1 equals f of A sub n instead of dA by dt equals f of A, would the bifurcations and the stability be the same? And the answer is no, they're not. They're related, and you can derive them, but they're not quite the same. And I'd be happy to sit down with you and walk you through the conversion from one to the other, because they are related to each other. But it's a little bit too much math to do in the lecture today.
Yeah, thank you for that. Anyway.
If you're interested, the textbook that I recommended, that Essential Mathematical Biology textbook of Britain, has a very good chapter on the basics of stability of iterated maps. And I should say that the very first papers I ever published were on iterated maps. 30 some years ago, when I was a grad student. Actually, experimental papers, not theoretical papers, but the experimental papers. And so iterated maps do show up in the real world in experiments, but they're not things that we're going to run into so much in the kind of computational biology we're doing here.
One question. So when you think about physics, then you have liquid, solid, gas. These are our phases, and this will be the fixed points, right? So how is the map then there? Is this related? I don't know.
So Elmer asked the question, we talk about phase diagrams in physics when we talk about things like solid to liquid to gas transitions for a material. And here we're talking about bifurcations and stability for dynamical equations. And he asks, are they related? And the answer is absolutely they are related. And a phase transition is a kind of bifurcation. Now, there are many, many different kinds of phase transition, material phase transitions, some of which correspond to bifurcations that we're going to be addressing in this class, and some of which are more complex. But the basic analysis, for example, perturbation analysis that I was talking about, is something that one does when one does the theory of phase transitions as well in physics. So there's quite a rich mathematics and theory about these things. Again, in a class like this, which is not fundamentally a math class, I can only touch on a few of these ideas, but I certainly appreciate people's interest in these questions. Okay, so I would like people now to do a little bit of an exercise, and I hope people have that. I think it was lecture 7 a long time ago, that notebook that we had. And if people don't have it, Hayden maybe, oh, he says he's out, stepped away for a second. People maybe can share that together. I gave you a notebook that actually we solved this problem, and you plotted the time series to find the fixed points. And then you iterated by changing the parameters, and plotted the fixed points as a function of the parameters. And I had sort of thought people might be in person. I'm not quite sure how to do this effectively with one person in the room and 4 people online. I guess I could create some breakout rooms. But I would like people to go, and I would like people to plot the bifurcation diagram for this ordinary differential equation here as you change K Basel. So what you would do is you would evaluate, you'd take K Basel, you set the value of K Basel, you try a bunch of different initial values of A, you run them forward in time, you find the steady state, and then you plot the steady state versus K Basel. Then you increment K Basel, and you do it again, and you plot that as a series, not a time series in this case, but a plot of the fixed point versus K Basel. And I had assigned you something which I think I asked you to do a couple of weeks ago, but we didn't do in class together, which is I'd like you to find the unstable fixed point too. And that seems initially to be quite hard, because if you start somewhere near a stable fixed point and iterate, you get the value of the stable fixed point. If you start near an unstable fixed point and run forward in time, you move away from it. And that actually tells you how to find the unstable fixed points. If I simply reverse the direction of time. In this case, instead of reversing time, I say f of a, that is k Basel plus a to the h over such and such minus d1 times a. If I put a minus sign in front of everything on the right, and then run that forward in time, that's equivalent to running backwards in time. And so in that case, I will find the unstable fixed point. And so if you're going to do this in Tellurium, what you can do is you can multiply your entire equation, your entire rate equation, by a number, and you multiply by +1 if you're going to find the stable fixed points, and -1 if you're going to find the unstable fixed points. And so I'd like everybody to dust off the code that we had, or write it again. I already gave you. You have it. But I would like you to extend it to find the unstable fixed point as well. And I think that this is one where perhaps having people, you're welcome to try this on your own, but if you would like to work in a little group, I can set up some breakout rooms for you. So tell me how you'd like to work on this. And I will give you 10 minutes or 15 minutes to do this, and I will expect everybody to have a result. So this is actually pretty important. So what's the best way to help you guys work together to come up with this result? How many breakout rooms do you need? Do you want to do 2 groups of, we have 5 people, so do you want to have a group of 2 and a group of 3, or do you all want to do it together? How do you want to do it?
I mean, if we all did it together, we get it. I'd hope.
Okay. So what should I do? Should I create a breakout room where you work together and you come back here? Should I just unshare the screen and let you guys talk together? How do you want to do it? Does somebody want to fire up a notebook and you can all talk to each other about how to work on the notebook? Is that a good way to do it?
Well, now that I think about it more,
It might be
also-
too hectic with too many people.
yeah,
I
we
think
should
just
we
2
split
groups
it.
is probably good.
Okay, so let me create a breakout. I'll create 2 breakout rooms, and I'll let you choose. And if you forget what the assignment is, I can put the assignment in the chat, or you can come back here. So, Pedro, how do you feel about this as something does everybody have the old code that I think I've seen that code a couple of times because people have been showing me notebooks that look like they have that code in it.
Yeah, maybe I don't understand, but is this the correct notebook? Or I might just.
So I gave you a couple of weeks ago when we did this problem, I gave you a notebook which actually had this code in it to find the stable fixed points. What is missing from that code is the unstable fixed point finder. So to make this a tiny bit more than just run the code I gave you 3 weeks ago or 4 weeks ago, I'm asking you to extend that code to find the unstable fixed point as well. Okay? And so why don't Pedro, are you there?
Yes.
Okay. How are you with this as a problem? Do you have the old code that we had?
The one that Hayden placed in the chat?
I shared with you a notebook a long time ago, a Colab notebook that calculated the stable fixed points for this specific problem.
Yeah, it's not. I don't think it's the one that he shared. So I think let me try to find it.
I really encourage everybody to continue to keep the notebooks you've got and to work with them. Don't start from scratch every time you do something. So while you guys are working, I can dig up the notebook and find it for you. But you should have it. In other words, as a matter of policy, everyone in this class, when you work with a notebook, you should save a copy of it for yourselves.
Do you
So,
know what the name is?
so you guys go work, and you have to decide. I want two people in room 1 and 3 people in room 2. And you work on it. And while you're doing that, I will go find that notebook and I'll put that in the chat for people. Okay. But why don't you get to work on your own? Regenerating the whole thing from scratch takes about 10 minutes. So if you don't find it, it's not the end of the world, but I would like you to try working on it together. Okay. So I cleaned up the notebook a little bit. This cell here, I've labeled it plot bifurcation diagram. This is the cell that you want to work with. You're going to have to change the reactions to be the ones that are at exercise 12.1. That's a little bit simpler. And you'll want to scan K Basel, which is given here, where we do line 21, which is where you scan the parameter. So I would first plot K Basel, change K Basel. And then after you've done that, your assignment is to find the unstable fixed point as well. So the code is there for it. Okay.
James, how do I run the reaction backwards?
Sorry?
How do I run the reaction backwards?
Well, you have to figure that out.
Okay.
It takes exactly one line of code, so you can
Yeah,
figure.
yeah, but the result doesn't make sense. I mean, I just inverted the signs
That's right. That's
of
all
the
you
problem.
need to do.
Yeah. Oh, okay. Okay, so maybe I didn't interpret the results correctly.
So the question is, if you start with the wrong initial value, it'll diverge.
Maybe. Yeah. Let me see.
So you have to decide where to start, what initial conditions to use that are correct. Okay. So I hope that this notebook should now be available. I'll do this. I'm going to not share the screen, but I'm going to do this myself while you're doing it. So I think, okay, everybody, you could stay in the main room too if you'd rather, instead of going to a breakout room, talk together. But you could work in the shared notebook too, because with Colab you could edit, you could work in the same notebook as well. Okay, give it a shot.
So Ibrahim,
Yep.
I think maybe this one fixed point that you had was just the one that was diverging. That's why we had all these different points, and that's why we not got it.
Maybe I'll try if I can
I'll
screen
check them out,
share.
but just was coming to my mind.
Yeah, that might be it. I'll screen share again and just work on the section that he had brought originally. I think it's this one. This. Let's just change this to what we had here.
Okay.
This should be. INNP.
Okay. Oh, sorry.
Okay, so here we're plotting for A1 in this range. Changing be a Basel rate or assigning a Basel rate, simulating. Now we're appending the Basel rate and RRSV 0. Can someone remind me what this is? Elmer or William, do you remember?
Sorry, I was working on code. What this one?
RRSV
So
0.
RR was your
This is
the
the
result, and SV this takes the as far as I know, this takes the first column out of your result. If yeah, so this SV or RSV will be I mean it takes for sure the first element from a list, but I don't know anymore what the SV is.
I thought it's a steady state.
Is it?
I think
Steady
so.
state's value, and then it's the first.
Yeah, steady state.
Okay, so it takes out the steady state value and appends it to the list. So in the end, you have a list of steady state values. So your L1 will be an array of steady state values.
okay.
Can you just print our RSV to see what the whole thing is, not only the first element?
Okay, yeah, so here we're just plotting. Yeah, this is it then. This is just plotting the Basel rate.
Basel, yeah.
And then the steady
Steady
state
state of A.
of A.
That makes sense. Yeah, that makes sense.
But we have to find the unstable fixed point also.
So what he said, what Cem said is that we have to reverse the. So we most probably have to go to the formula of where we the code, and then we have to multiply by -1.
Yes, I think you're right. Let's do this in another cell.
So Zoom is just a pleasure. I hate Zoom so much. Sorry. It's annoying with the small screen.
It didn't find a steady state, it looks like.
Wait. You Oh, never mind. Oh. I don't think it's gonna work because of what the negatives in virtual double.
Could just do.
What if we just put one negative sign?
Yeah,
Or
same
can we not
thing.
just?
Too much work.
Can we not just reverse the sign?
You want to do just for this one?
You have as well to do this over all of the KPs. The moment you don't change the KPs. Oh, change to B. Okay, yeah.
Maybe I just do this part by -1. Not including the Basel rate.
I just can't even get mine to run.
Just failures.
Okay, here we go.
I feel like just flipping the arrows around should get the reverse of the reaction.
Yeah.
But it's not arriving at any steady state.
Huh. Pedro, do you have anything?
I am doing Monte Carlo stuff here. Try and error.
Got it. Yeah, it's kind of sad from us.
Not reach T out 20,000 steps.
Why are we loading?
I think I found it, but I mean I don't trust my results.
How did you flip the reaction? Did you just change the arrows or did you just multiply everything by -1?
I multiplied by -1, but I don't know if it's right. Maybe.
Interesting.
Oh wait, yeah, I actually wait. I think I got it. Yeah, wait. What did you get? Like a lot. Like
What did you do, William, to get that?
okay, I don't think I'm right. I just got a line. I just stuck negatives in front of negative the Basel rate and then a to the h, because you don't need negatives on everything. You just need a negative there, and then yeah.
You didn't
Oh.
get this error?
No. Wait, let me.
There we go. Okay, good.
No, I didn't get that. Wait, let me.
You want to share your screen?
I mean, yeah, it's. I don't know. I mean, I think this must be wrong, but
If you're getting something, it's got to be better than what I have.
you know what I mean. Oh crap! I have so many screens. Wait. Okay. Yeah. Like, I get this.
No, this is wrong, man.
Yeah, this is so wrong. Yeah, that's what I thought. Like,
I got a bizarre stuff here. So the stability is a problem because you're reversing in time. So we are probably, since we are not like starting exactly at a good value of A, the likelihood that you explode is very high, unless you really start at your endpoint. As I don't know the endpoint exactly, I am just saying if the value of A is above 100 or below 0, don't even give me. And I am hoping something between 0.1 and 6 works. That's it. And then I get this weird shaped graph here. So the unstable equilibrium point, as k increases, would diminishes. However, I don't understand why it's a closed loop. Maybe it's a problem with plotting. I have no idea actually. But I mean, my instinct tells me it's wrong.
Plotting
I'm
them on top of
sure.
each other maybe is a little bit more work. Were you able to find the three?
I found one of the three.
Okay, I think we should probably move on. I put it in the notebook. Let me show you my code maybe. Or how far did you get? You get it?
I think I did. I'm unsure, but
Let me just show you how I did it, and then I wasn't very elegant, but we can take a look together.
I think
Can people see that all right?
yes.
Okay. So what did I do? I defined my reaction here, and I added a variable called direction, which is forward and backward direction. So normally it's 1. And if I want to find the unstable fixed point, I make it -1. Okay. Now I load my model, and then I wrote some code that will plot the time series just to make sure that things are well behaved. And then here's what I do. I'm going to scan my parameter. In this case, it's going to be, actually this A1 isn't being used at the moment. I could have gotten rid of that. I'm going to scan I, which is going to be controlling my parameter, between 0 and 1, small steps. I reset the value. I set K Basel equal to I. I start with a small value of A. There is no B here. I run the simulation for a while. I set A low equal to the outcome when I started with a low value. I reset it. I reset my value of K Basel. I start with a high value of A. I run for a while. I set A high equal to that. I reset. I set my K Basel again. Now I set my direction to be backwards. I start somewhere in the middle because I know it has to be between those two limits. I really should pick something that's in between K low and K high. So if I want to be a little bit better on this, I could do this would be 0.5 times A low plus A high. Then I'm guaranteed to be within the range, which is better. I simulate now, and now the simulation could diverge because when you run backwards in time, it's unstable. So I check if the value of my output is between 2 is positive and between 2. If it's well behaved, I add it. If it's not well behaved, I hide behind A high, which I already have, and I stack it. So let's see whether that works. I get that. So the lower branch is the stable lower value. The middle branch is the unstable value. And the upper branch is the stable upper value. Okay. Why doesn't everybody try downloading the new version of the code and see if you can get it to run for yourselves? So it's the only trick that you have to do is that you have to check to see whether your value diverged or not. Any questions about that? Does that make sense to people? So all you had to do was two things. You had to add the minus sign. You had to add the minus sign here for direction. And then when you ran in the reverse direction, you had to check whether the value was divergent or not. Because if the value is divergent, you want to throw it out. Okay? Does that make sense, Pedro? Does that make sense to you?
Yes, I think I did the same as you, but not with but I did step by step. I mean, first I ran, then I chose the value in between, then I changed the code, and then I run again.
Right.
So you did it automatically. I did it like playing around.
Okay. Well, that's okay. I mean, there are a lot of different ways to do it. I did want people. I don't want to take. It's already 6:30, and we're going to run out of time again. So I want to keep going, but I did want people to try thinking about this a little bit because it's a little bit more interesting maybe to think about this problem than some of the things we've done. All right. And I have to say, I did want to have those unstable fixed points on my plot, so I wanted to make sure I had it. All right. So your next assignment is what if we change D, the decay rate, instead of the K Basel? That effectively pivots our curve downward as we make D1 bigger. And so if we make D1 very small, we pivot our curve upward. We pivot our curve counterclockwise. around 0. If we make D1 large, we pivot our curve clockwise. And so what happens? If we pivot our curve counterclockwise, we make the decay rate smaller. That upper fixed point moves to the right. And in fact, if we make D1 0, it becomes infinite. And the unstable fixed point also moves to the right. And you'll see that that unstable fixed point will disappear if we make D1 too big. I mean, too small. If we make D1 too bigger, then we're pivoting the curve clockwise. The upper stable fixed point moves to the left. The upper unstable fixed point moves to the right, and they collide. And eventually, the only stable fixed point we have is 0. So why don't people take the code that I just wrote? You should have access to it. I'll share it with you again. And just change the scan so that instead of scanning K-Basel, you are now scanning D1. And give me the bifurcation diagram for that. Is that okay?
I mean, I just went from changed D1 from 0.3 to 2. And
that looks perfect.
Yeah, I mean that's it, right? Yeah.
Yeah. That's what I was saying. It doesn't actually take that much to do.
Well, no, I was just confused. I just had this for a while, and I was just maybe I should have said something a while ago. But yeah, I was just confused.
Seemed too easy. Too easy.
Yes.
Okay. Well, sometimes I'm afraid sometimes that the problem is that I assign things that people think are more difficult than they are, and so that people get confused because it's too easy. So thank you, William. Okay, great. Okay, good. So by all means, if you're having trouble with this, ask William for advice. His solution is more or less the one that I put in my code when I saved it just now. The one thing that's a little bit tricky is you get that little extra line where the bifurcation disappears. You get a little bit of a line that draws where it shouldn't draw between the end of the where the fixed point disappears. It draws a line down to the x-axis. But we could play around with the plotting a little bit so that line is not there. For purposes of understanding what's going on, that's fine. Great, good job. Okay. So we've seen the bifurcation plot now for increasing D1. And what we see is if D1 is very small, if D1 is 0, the value of A goes to infinity. If D1 is small, the value of A is very large. As we increase D1, that large value comes down. The value at 0 stays 0 if K-Basel is 0. And the unstable fixed point gradually goes up until it collides with the decreasing value of the upper fixed point. And they annihilate each other. And if the decay rate is too high, you get 0. All right. So what I'd like to do in our remaining time today, and I want to make sure everybody can see the slides again. William, can you see the slides?
Yep.
Is we've seen a couple of different kinds of bifurcation. And I want to ask the question, can we categorize the kinds of bifurcation that we have? How many are there and what do they look like? And there's going to be a little bit of math that I may skip over. I'll leave you, the details will be in the deck if you want to dig through it in more detail. But it's actually possible to categorize all of the one-dimensional bifurcations. We can actually also categorize all the two-dimensional bifurcations. And when we get to 3 dimensions and higher, it winds up they only add 1 or 2 more. So there are not actually that many different bifurcations that are possible. for continuous differentiable functions. So in one dimension, you can determine the types of bifurcations essentially by what are called the reduced or canonical forms of the equations. That is, the lowest order terms of the Taylor expansion around the fixed point. And the reason the math is a little bit finicky, it's not difficult, but reducing things to their canonical forms means doing a little bit of algebra. But conceptually, it's pretty simple, but the algebra is a bit different for each one. There are 4 possible bifurcations in one dimension. We've seen some of them already. They have some rather, I don't know, descriptive names. The first one is called the saddle node or tangent bifurcation. The second one is called the transcritical or exchange of stability bifurcation. That second one is sort of trivial looking. And then the third one is what's called a pitchfork bifurcation. You'll see why in a minute. And the fourth one is what's called an inverse pitchfork bifurcation, which is one that you also run into occasionally, but isn't necessarily the most exciting. The ones we've seen in the example we did were both saddle node or tangent bifurcations, as it happens. Okay, so what are we going to do? We're going to adjust lambda. We're going to rework these equations so that the bifurcation happens when lambda equals 0. And we could always shift lambda by a constant to recover a general parameter. And we're also going to adjust the values of x so that the fixed point is at 0. And so what we're trying to do is take an arbitrary functional form, f of lambda and x, and we're going to slide it around so that the fixed point is at 0, and also the value of the parameter at which the bifurcation happens is at 0. And that's where the little bit of extra algebra comes from. So now let's assume that x star is equals 0 as a fixed point. And we're going to Taylor expand f of lambda, and we'll get a0 plus a1 times x plus a2 times x squared plus a3 times x cubed, and so on. The lowest possible order for that would be the first 2 terms, a0 plus a1x. And the problem with that is that it doesn't change its slope at 0. It's not tangent to 0 at 0. And so there's no bifurcation. The next one that we get would be f of lambda comma x is a0 plus a2x squared. It's the next one. Okay. And that one is going to have a bifurcation if a2 is less than 0. And so we can rewrite this by dividing through by a2. And so then f of lambda comma x has the form lambda minus x squared. So clearly, in this case, if lambda is 0, minus x squared is always negative. If lambda is positive, then there's some finite value of x at which lambda minus x squared is 0. And so we can plot that here like this. We say dx by dt is equal to lambda squared minus x squared. And we get 2 fixed points. We get a fixed point where x is equal to lambda. And we get a fixed point where, sorry, I've written this in a funny way here. I've got a fixed point where x is equal to plus or minus the square root of lambda. And if lambda is negative, there's no fixed point. And so this is actually corresponding to what we had just done, where we have a stable and an unstable fixed point that come together and they collide. And then as we keep changing our parameter, there is no more fixed point. It disappears. And so the example that we just did was a case of this kind. We could flip it so that the increase, in this case, as we increase lambda, we get the fixed point. We could flip it so that we get, as we decrease lambda, we get the fixed point as well. So that was the first one that we get. The next one we get, the first one we had was a0 plus a2x squared. The next order one would be if a0 is 0, then we have a1x plus a2x squared. And in this case, again, we divide x by the square root of a2. And we're left with f of lambda comma x is lambda x minus x squared. And so in this case, we have dx by dt is equal to lambda x minus x squared. And in this case, the sign of the slope changes when lambda goes from plus to minus. So what are our roots of this? If x is 0, clearly dx by dt is 0. And if x equals lambda, lambda times lambda is lambda squared minus lambda squared is 0. And so again, we have two fixed points. The trivial fixed point, which is x equals 0, and the fixed point where x equals lambda. In this case, if lambda is negative, the fixed point at 0 is stable, and the fixed point at lambda is not unstable. If lambda is positive, the fixed point at 0 is unstable, and the fixed point at lambda is stable. And so before we had two fixed points that hit each other and disappeared as we changed lambda. Here we have a stable and an unstable fixed point. They hit each other and they exchange stability. The stable one becomes unstable. The unstable one becomes stable. And so the number of fixed points here doesn't change. Whether they're stable or unstable, the number of unstable and number of stable fixed points also doesn't change. But the stable one becomes unstable and the unstable one becomes stable. This isn't the most exciting thing to happen, but it's one that happens pretty often when you write complex first-order ODEs. That's all the ones that have first-order x squared. The next one that we have is the x cubed. And here we have to divide through by a3 to the 1/3 minus 1/3. So not as pretty. And we're left with f of lambda comma x equals lambda x plus or minus x cubed. We can pull out the x to make it look a little simpler. We have x times lambda plus or minus x squared. And so here, our equation is the form dx by dt is equal to x times lambda minus x squared. And we want to know what the roots are. Well, again, clearly, if x is equal to 0, then dx by dt is 0. So x equals 0 is always a root. If lambda is positive, plus or minus the square root of lambda is a root. If lambda is negative, that second one doesn't have a root. And so what we have now is if lambda is negative, we have just, well, we could have a complex root, but here we're insisting that x be real. Elmer says, what about a complex root? If we're in 2 dimensions, the complex root actually is the oscillatory solution. This would correspond to what's called the Hopf bifurcation. in 2 dimensions, but we haven't gotten to that one yet. We'll do that next time. Okay. But in this case, you have a stable fixed point at 0 for lambda negative. When lambda becomes positive, the stable fixed point becomes unstable, and we get a pair of stable branches at square root of lambda and minus square root of lambda. And I think you could see why it's called a pitchfork bifurcation, because on the left-hand side, it looks like the handle of the pitchfork, and on the right-hand side, it looks like the center, the unstable branch is the center tine of the pitchfork, and then you have the top and the bottom branch are the other branches. You'll notice this actually looks very similar. If we go back, this looks very similar to this one, where we also had roots at square root of lambda and minus square root of lambda. But instead of nothing on the left and a stable on the top and unstable on the bottom, we have 0 on the left, stable on the top and stable on the bottom, and unstable in the middle. And so these are related to each other. These bifurcations are related to each other, but not quite the same. And the last one is simply reversing the sign of what we just did. And in this case, we have 2 unstable fixed points at plus minus square root of minus lambda. And we have a stable fixed point at 0. And these collide and leave us with an unstable fixed point at 0 on the right. And this inverse pitchfork bifurcation doesn't look really that exciting. What it would look like if you were just doing the simulation is that you would have a stable fixed point disappear. And typically, you don't study this one on its own. This is sort of the leftovers when other things are happening. But it's there, and it does happen. And that's it. Those are all of the possible kinds of bifurcation that can occur in 1 dimension. And I realize I went through that a little bit fast. I don't think that you need to have the math is not that critical for the moment. What's critical is to know that you can classify the 1-dimensional bifurcations, that there are only 4 kinds, and that they have these basic sets of structures. Elmer, go ahead.
Yeah, can I revise too? I'm not sure if I answered. So are you going down the terms from the Taylor expansion to find the different types of the bifurcation?
Yeah, so you could ask why aren't there more? And that's a legitimate question. So if I Taylor expand F to begin with around 0, the Taylor expansion of X in X is constant plus A1 times X plus A2 times X squared plus A3 times X cubed plus other stuff. And what I'm asking is, if I assume that the value of X is small, the lowest order terms are going to be the biggest ones, because if X is 0.001 and I have X to the 4th, then X to the 4th of 0.001 is a very small number. And so if A0 is not 0, then it's going to dominate. If I have A0, 0, then A1 term will dominate. If I have A1, 0 and A1, 0, then A2 term will dominate, and so on. And so what I'm trying to do is I'm trying to look for the lowest order in X terms and ask how do they bifurcate. And what I have not shown mathematically is why there couldn't be additional ones for, say, X to the 5th or X to the 7th. And that one you'll have to look up. The proof of that is not so trivial. But what we've done is we've essentially said it's a little bit like a Boolean thing where we say well, the first digit is a 1. What are all the possibilities? Well, the first digit's a 0, so the second digit's a 1. What are the possibilities? And so we're walking through those. And I've done that in a bit of a hurry because I don't want to take too much time on this. For here, as I say, if you want, I noticed a typo or two in the slides too. So, okay, but there is one more thing that I wanted to talk about. Oops, I've got to come back here. There is something that is rather pretty. There's a nice theorem, and I think that's where we'll pretty much end for today. We don't prove theorems very much, but the statement that you always get a fixed point in a one-dimensional ODE or a divergence seems awfully simple. And you could ask the question, why can't I get an oscillatory solution? I've said already that you can't get oscillations for a simple first-order ordinary differential equation. And there's actually sort of a fun little theorem. It's a little bit like the ancient Greek theorem that the square root of 2 is irrational. I remember our math teacher, as a freshman in high school, our math teacher gave us the little proof that the square root of 2 is irrational. So yeah, the way you prove it is you say, suppose it were rational, so it were P over Q, and you assume that there are no common denominators between P and Q. It's in lowest order. It's been reduced, and then you show that it can't be. So it's a contradiction proof. That was actually a proof that was known to the ancient Greeks. They didn't do a lot of, most of their proofs were very geometric. They didn't do a lot of algebraic proofs, but that was one they had. So this proof is a little bit like that. So let's suppose that our time series x of t is periodic. So that means that x of t is equal to x of t plus tau. There's some period, right? That's what we mean by periodic. Okay. And we know that dx by dt is f of x. So let's multiply on the left-hand and the right-hand side by dx by dt. It's actually very similar to the proof that the square root of 2 is irrational. That's why I was mentioned. Okay, so then the left-hand side becomes dx by dt squared. Nothing funny going on there. The right-hand side is f of x times dx by dt. So nothing up my sleeve. I now am going to integrate both sides from t to t plus tau. I'm going to integrate over one period of my oscillation. Again, nothing terribly tricky going on. I have the integral from t to t plus tau of dx by dt quantity squared, integrated by dt. On the right-hand side, I have t to t plus tau, f of x dx by dt times dt. Well, let's see what happens to the right-hand side. The integral of dx by dt, usually you can't cancel dt's, but in this case, inside of an integral, you do. It's a change of variables. And so you wind up with the integral of f of x from x of t to x of t plus tau. That's just a change of variables. But x of t plus tau is equal to x of t because it's periodic. So I have the integral from x of t to x of t of f of x, which is 0. So the right-hand side of this equation is 0. But the left-hand side, dx by dt squared, is always greater than or equal to 0 because it's the square of a number. So the integral from t to t plus tau of dx by over dt squared dt has to be greater than or equal to 0. But we know the right-hand side, from the right-hand side, that that's equal to 0. So the only way dx by dt quantity squared integral can be 0 is if dx by dt is 0 everywhere. So if dx by dt is 0 everywhere, f of x is 0. So the only periodic solution that you can have is x as a constant. So there's a proof that there are no periodic solutions in one dimension. If you have an aperiodic, you said, well, what if it were chaotic instead of periodic? Even if it's chaotic, there have to be 2 time points at which it has the same value. And you can do exactly the same thing between those 2 time points. And so this actually gives you a fundamental result, which is that a one-dimensional first-order differential equation cannot have a periodic or chaotic time series. And so I think that that's, again, I try to avoid doing a lot of mathematics, but that one is a simple enough proof, and it's an elegant enough result that I thought it might be fun for you to see it. Now, of course, in the biology we're almost never dealing with things where there's only one variable, right? A excites A is about the only thing we're ever going to deal with there. We're always dealing with A inhibits B, B inhibits A does something else. We're dealing with much more elaborate systems. But the basic underlying underpinnings of this are still nice to know. Okay, so I'll just start us off, and then we'll come back to the higher dimensional dynamics later. We've done the basic stability for one-dimensional systems, and now we want to begin to think about how we're going to determine fixed points and stability and bifurcations if we have a lot of equations. And so suppose that we have, I've written, the notation is a little bit different from the one I had before, but we have dn1 by dt is some function of all our species concentrations. dn2 by dt is equal to some function of all the species concentrations, and so on. So we'll write, again, dn by dt is equal to some vector function of n. Okay. And the stability is going to be defined just the way we did it before, which is a fixed point will be a set of concentrations for which f of that is 0. And as before, if we perturb away from that initial position n star, if we come back, we'll say our fixed point is stable. And if we move further away, we'll say our fixed point is unstable. The difference here is that before, the perturbation was in just one variable, and so all we had to look at was the value, was epsilon getting bigger or smaller. Here, epsilon is a vector, and so it could be changing both its length and its direction. And for stability, we need its length to be decreasing. And so we'll come back next time to understanding what that is. To get some insight into that, we will use the thing that we've kept talking about but never really defined, which are the ideas of null clines and flows. And we've already plotted these phase portraits of systems. One of them is in the notebook here.
Null clines equidistance.
And okay, so Elmer asked the question: are null clines equidistant? And so the word cline is one that mathematicians and geographers use. There's actually, if you drive from here to Chicago, there is actually a Cline Avenue. And it really is Cline, as in this Cline. not Cline is in the name of a person. So a Cline is a set of points that obey a particular rule. And so when you do Clines in a graphic plot, you are typically plotting the set of points where, for example, the function has the value of 10. A null-cline is a set of points where a function has a value of 0. And in particular, if we have a two-dimensional or a higher dimensional system, a fixed point is when all of our rate equations are 0. F1 is 0, F2 is 0, F3 is 0. The rate of change of x1 is 0, the rate of change of x2 is 0, the rate of change of x3 is 0. The null clines are the places where only one of those is 0. So suppose we have two equations. We have an equation for x and an equation for y. The null cline for x is where dx by dt is 0. But that doesn't mean that dy by dt is 0. The null cline for y is where dy by dt is 0. That doesn't mean that dx by dt is 0. Where the null clines cross, dx by dt is 0 and dy by dt is 0, that's a fixed point. So null clines cross at fixed points.
When you have an equation system, it's just one equation.
So it doesn't make sense to talk about a null cline for a single variable. For a single variable, the null cline is the fixed point. Now, why would you like this? And the main reason you like null clines is not so much for analytic calculations or even for numerical calculations. It's really to give you a graphical way of understanding the global, not the local stability, but the global stability of the system. So a null cline is a set of points where the right-hand side, the rate of one of the rate equations is 0. And the nice thing is that in a null cline, you know the direction in which the flow is going because it's perpendicular to the direction in which things are 0. So if you have along the null cline for x, things are moved, change, the y value can change, but the x value can't change. So you know the direction of flow on a null cline. Null clines cross at fixed points. So now, and I may take an extra minute or two here, because this is something that was asked about at the beginning, and it took us a little bit longer than I had hoped to do the first exercise. And I do want to show you this because I promised that I would show you this. So I hope you'll give me just a couple of extra minutes here. There's a simple predator-prey model called the Lotka-Volterra equation. And the Lotka-Volterra equation, V is the prey, and U is the predator. And actually, did I get that right? Or is it backwards? Let's see. I have to remember that. I'm going to ignore that question for the moment because I don't remember. I think I got it backwards. Okay. And we're just going to write this as two differential equations. We're going to say that the du by dt is equal to u times 1 minus v. Now, v is the prey. V is the predator. U is the prey, and V is the predator. So U grows exponentially if there are no predators, and the predators eat the prey, and so the more predators I have, the less U I get. dV by dt is equal to alpha times V times U. The predators only can reproduce if they're prey for them to eat. This has two clear fixed points. If U and V are both 0, all of these things are 0. And if they're both 1, they're both the rates are both 0. In this case, the null-clines are easy to determine. What is the u null-cline? I set du by dt equal to 0. That means u times 1 minus v is 0. And there are two possibilities. If v is equal to 1 and u is equal to anything, that gives me a line, it's 0. Or if u equals 0 and v equals anything, I also get a line. So I actually have two lines. There are two null-clines coming out of this. So all points that are of the form u comma 1 or 0 comma v are u null-clines. I'll give you a second to think about that. The v null-cline is when dv dt equals 0. That means av times 1 minus u equals 0. Well, if v is equal to 0, that's a null-cline for that. Or if u equals 1, that's a null-cline for that. So all points of the form 1 comma v or 0 comma u are v null-clines. Got that? That's actually rather a lot to digest in one. We're just drawing straight lines. There's nothing very complicated going on, but there are a couple of cases to worry about. Okay.
Well, I had a question.
Sure. Go ahead.
Yeah, with the du dt, dv dt,
yeah,
those equations, is that just from the model?
You mean why did I write? Where did those come from? Why did I pull them out of a hat?
Yeah.
So for the purposes of today, I've just pulled them out of a hat. These are Lotka Volterra was working. Volterra was working in the 1910 about and trying to come up with a simple mathematical theory of why you had oscillations in the populations of predator-prey systems. If you had foxes eating rabbits, why did you have oscillations in the populations of foxes and rabbits? And so this was actually one of the first simple mathematical models of population dynamics. As a model of predators and prey, it's defective in an awful lot of ways. It has all sorts of problems with it, but it's algebraically the simplest possible model you can write down that has oscillations, and so people still use it. The main reason the main defect of it is also the reason that it's simple, which is that it's what's called bilinear. It's linear in u. U appears as just u, not u squared or 1 over u or something like that. And it's linear in v. V appears just as v, not v squared, 1 over v, 1 over 1 plus v, or something of that kind. And that linearity means that it's very simple to work with. It also means that it has some properties that are not so pretty. And I'm going to run out of time, but I'm going to just show you what you need to do with this. So I can now draw my lines. Remember my lines. The u null-cline is v equals 1 or u equals 0. And so the u null-clines here are in green. I've drawn those two lines. The v null-cline is u equals 1 or v equals 0. I've drawn those in red. You see that on the picture, all right? William, can you see that? Where the null-clines cross, that is where the green line crosses the red one, I have a fixed point. So I have a fixed point at 1 comma 1 and 0 comma 0. And now what I'm going to do, and this is how you deal with null-clines and why people like them, is that I know when I cross the u null-cline, my lines have to be vertical because I'm not changing u. So I can draw little vertical lines across my u null-cline because my flow has to be vertical. I don't know whether it's up or down, but it's got to be vertical. And it's important to note that these lines are always vertical. They're not perpendicular to the null cline. So if the null cline were at an angle, they'd still be vertical lines. Similarly, I'm going to skip now. For the second one, for the V null cline, I have horizontal lines. And so now I know that along that vertical line, that red vertical line, the flow is horizontal. Along that horizontal green line, the flow is vertical. Now, what I have not yet done, and I won't have time to do today, is ask the question, what direction are you going? In other words, when you cross that horizontal line, are you going to the left? I'm sorry, when you cross the horizontal line, are you going up or down? When you cross the vertical line, are you going to the left or the right? And what's nice is you can find any point where you know the direction of movement. And when you cross V null cline, the direction of movement in the V direction flips. When you cross a U null cline, the direction of movement in the vertical direction flips. And that means that if you know the direction of movement in one point anywhere here, you can then walk across. Each time you cross green null cline, you flip the vertical direction. Each time you cross a red null cline, you flip the horizontal direction. And then without doing any math at all, I can draw the entire flow for the entire field. And so once I can draw my null clines and evaluate the flow at a single point, I can evaluate the flow everywhere in the system without having to do any more math. And that's why it's considered a very powerful technique. Of course, if I can run a simulation to do the thing everywhere all the time, I don't need to do it by hand. But there's something very satisfying, I think, in helping you really feel that you understand the system by being able to say, gee, if I draw these lines and I evaluate what's happening in a single point, it tells me what happens everywhere in the whole space. And so that's the 10-minute version of null clines. And now we'll come back next time. Now that you've seen them once, we can come back and begin to think about them next time more seriously. Okay? Elmer, what's the question?
You said that they are horizontal, vertical, not perpendicular, but because in this example they are perpendicular.
Right. Let me just flip to one that where they're not so trivial. There's a set of null clines that are not so trivial.
They are not. Okay. We will come to that. Yeah.
But in this case, the null cline, the vertical, the V dot null cline is a steep line cutting along the middle. The U dot null cline is a sigmoid, is a cubic. And that one, the Fitsu-Nagumo, is a particularly interesting one because that's actually an equation that's used to explain how neurons behave.
So the only thing that changed that you have an A term there, right? Yes.
So you'll see it's cubic. This one happens to be cubic in U. I have U times U minus A times 1 minus U. So it's actually U cubed. So it's a cubic equation. Okay. So I ran over a little bit. Are there any other questions before we break? William, go ahead.
Yeah, wait, so
you should just break in. You shouldn't be so polite.
okay, sorry. But could you go back to, I guess it doesn't really matter, but for the flow, is
Yes.
that what does that mean? Is that over time? I think I read over time. That's how that point is moving over time. I don't understand.
So this is what we were doing for a couple of weeks now, where we plotted we say, suppose the initial value of u and v is a half comma half.
Yeah.
Now, in this case,
Oh,
u will increase and v will decrease.
okay, okay.
And then when I get to u equals 1, v equals a quarter, u will continue to increase, but v will start to increase again.
Okay.
Because it never finds, it will be periodic because it always writes around.
Right. In this case, you'll see that the populations will go around in circles,
Doesn't find a fixed point.
and there's no fixed point.
is
Right.
Got
periodic.
it. Okay, that makes sense.
It happens that the reason the Lotka-Volterra equations are sort of not pretty in some ways is because they're neutrally stable. The oscillations are neutrally stable. So once you go around a circle, you stay on whatever circle you start on, you stay on. In more complex models, there would be a stable cycle that would play the role of a stable fixed point.
But if you not have a stable cycle, then you are chaotic, right? Then you
No, in this case, you're periodic. For any initial condition, you're periodic.
but you have the same trajectory.
Yeah. It's a little bit the Lotka-Volterra is sort of a special case. That's why people like it because it's simple, but mathematically it has some properties that are not typical. And so again, I know that just the way, but I hope in a sense, just the way we did the basic stability very quickly at the end of last class, and I hope we went back and maybe consolidated that today. I wanted you to see this very quick, even if it's very quick, so that next time when we go over it more carefully, it won't be totally unfamiliar. And so I'm not expecting people to follow all of the details now, but I hope now, having seen it once in a bit of a hurry, it'll be more familiar when we come back and address it a second time. Okay? Okay, great. Thanks everybody for sticking it out today. I know we're a little bit over time.